Hey, welcome. Today we're going to do something pretty cool. We're going to take the food on your plate, think bread, pasta, potatoes, and zoom all the way in. I mean, way down to the microscopic level, to the actual chemical building blocks that power our bodies. Let's get right into it. So let's start with a question. What could table sugar and a potato possibly have in common? I know, it sounds a little weird, right? One is this sweet crystalline stuff, and the other is starchy and savory. But the answer, believe it or not, is hidden in the tiny, elegant molecules they're both built from. So let's just ask the big question. What is a carb, really? Now, if you ask a biochemist, the answer isn't just bread or sugar. To them, it's a very specific chemical blueprint, a kind of molecular recipe. So let's break that recipe down. Okay, I know that definition looks a little intense, but trust me, the idea behind it is actually pretty simple. All you have to do is imagine a backbone made of carbon atoms. Now, attached all over that backbone are a bunch of hydroxyl groups, which is just a fancy way of saying an oxygen and a hydrogen stuck together. And then there's one special chemical group, either an aldehyde or a ketone, that acts like a reactive little hook on the molecule. And that's it. That's the basic formula for every single carb.
So all the carbohydrates in the world, from the tiniest sugar molecule to the starch in a potato, fall into one of these three classes. The easiest way to think about it is with Legos. Monosaccharides are like a single Lego brick. Oligosaccharides, that's when you click a few bricks together, maybe 2 to 10. And polysaccharides are the giant, sprawling Lego castles. We're going to start with that single brick.
All right, let's meet that single brick, the monosaccharide. This is it. This is the fundamental unit, the basic building block that everything else is made from. But you know, they're not all created equal. The simplest way we classify these little guys is just by counting their carbons. A 3-carbon sugar is called a triose. A 5-carbon one is a pentose, and that group includes ribose, which is a super important part of our DNA. But the one we're really going to focus on is the 6-carbon hexose group, which includes the most famous sugar of them all, glucose. Now, it's really easy to draw these sugars as straight little lines, these open chains. But that's not actually how they hang out in our bodies. In the watery world inside our cells, they do something much more interesting. Remember that reactive hook we were talking about, the aldehyde or ketone? Well, it spontaneously reaches around and grabs onto a hydroxyl group on the same molecule, and it snaps the chain shut, forming this incredibly stable little ring. This ring shape is how you'll find sugars almost all the time inside our cells. And here's where it gets really cool. When that ring snaps shut, one of those hydroxyl groups on a very specific carbon can end up pointing either down or up. This tiny little flip creates 2 completely distinct versions of glucose. We call them alpha glucose and beta glucose. I know it seems like such a minor detail, but that simple up or down difference, it changes everything about the kinds of larger structures they can build. Okay, so now we have our single bricks, alpha and beta glucose. How do we start clicking them together to build something bigger? Well, for that, you need a special kind of chemical glue. And that glue has a name. It's called a glycosidic bond. It's a strong, covalent bond that forms between one sugar and another. This is the magic link that lets us go from single units to long chains and complex branches. So our first build is a simple one, just 2 bricks. When you link 2 monosaccharides together with one of those glycosidic bonds, you get what's called a disaccharide. Let's look at 3 really important ones you've definitely heard of. First up, we've got lactose. This is the sugar that gives milk its natural, subtle sweetness. It's made from one galactose brick linked to a glucose brick. And look at that bond. It's a beta bond. The entire difference between being able to digest milk easily and being lactose intolerant comes down to whether or not your body makes the right enzyme to break this specific beta bond. Next up is sucrose. You know this one as plain old table sugar, the stuff we get from sugarcane and sugar beets. This is what happens when you bond a glucose brick to a fructose brick. It's literally what makes your coffee sweet in the morning. And finally, we have maltose. This one's super simple. It's just 2 glucose bricks stuck together. You find it when you're malting grains for beer, and it's also the very first piece that your body breaks off when it starts digesting bigger carbohydrates like starch. And just notice that bond. See how it's an alpha bond? Tuck that away because alpha is going to be really important in a minute.
All right, time for the main event. We've seen single bricks and pairs of bricks, but what happens when you link hundreds or even thousands of them together? Well, then you get the absolute giants of the carbohydrate world, the polysaccharides.
These gigantic molecules, also known as polymers, are basically nature's way of storing massive amounts of energy in a really compact form. And by far, the favorite building block for these giants is our old friend, glucose. So, in our bodies, when we eat a meal and have a bunch of extra glucose floating around, we don't just waste it. We cleverly package it all up into this huge branch molecule called glycogen. Think of it as our personal, ready-to-go energy reserve, stored mostly in our liver and muscles. And what's amazing is that plants do the exact same thing. They just call their storage molecule starch. And this brings us right back to our first question. A potato is just jam-packed with starch, which is just the plant's version of storing thousands and thousands of glucose units for later. So when you get right down to it, they're both just long chains of glucose used for energy storage. But you have to look at that one key difference in structure. Glycogen is highly branched, while starch is only lightly branched. Now this is not a random detail. This is where the entire story really comes together. So why? Why does this difference in branching, this sort of molecular architecture, matter so much? Well, it all comes down to one thing: the speed of energy access. In glycogen, that's our energy reserve, you get a branch point about every 8 to 10 glucose units. In starch, it's way less frequent, more like every 20 to 30. This makes glycogen a much more compact, almost bushy kind of molecule. And this is the big payoff. This is why it matters. The enzymes in our body that free up glucose for energy can only work on the ends of the chains. So if you have more branches, you have way more ends for those enzymes to attack all at once. It means when your body needs energy, and it needs it fast, like if you're suddenly sprinting, it can unleash an enormous flood of glucose from glycogen almost instantly.
And that kind of leaves us with a final thought to chew on. The very shape of our energy storage molecule is optimized for rapid-fire release. Plants, they're stationary. They can get away with a less branched, slower release system like starch. But for animals, this highly branched glycogen is just this beautiful piece of biochemical engineering that reflects a fundamental truth about us. Sometimes you just need energy, and you need it right now.